Hi, my name is Hope. You should have already met me during the presentation talking about RosettaCM, and now we're going to do a quick walkthrough of this tutorial. So if you haven't already, you should open a terminal and go to the RosettaCM directory from the workshop. And from here, we're going to want to make a directory called My Model, and then go into that directory. And we're actually going to want to copy everything from the My Model directory into here. So go into Input Files, and we need to get the Fasta for our target protein. So we can go to the NCBI website and search for DRD3. And I would scroll down till you find the human one, and click Fasta. So now you can copy this text into a file called 3pbl.fasta. And we want to replace this first line with .3pbl and save that. So now we're going to alter a bit the sequence for modeling, but we want to save that full sequence for one of the later steps. So I'm actually going to copy this file into another file called 3pbl full.fasta. So now you want to go through and edit your Fasta file. So depending on if you downloaded that from the NCBI website or from the PDB, will be slightly different. But essentially, we don't want to model the N-terminus or this large ICL3, so there's going to be a lot of flexibility in those. And they're probably likely disordered. So the steps for that are listed in your tutorial. I actually have also that file prepared for you in the demos, so we can copy it over for the sake of time right now. So if you want to just take a quick look at that, this is what that looks like. Alright, so on to our template structures. So to find a template, we are going to use BLAST. So first I would just cat your Fasta sequence onto the screen and copy that real quick. And now we're going to want to go to the BLAST website. So what you do is paste your Fasta into here. You want to make sure that the PDB is chosen for your database, as we're looking for not just proteins that have a similar sequence, but also proteins that have a structure determined, so we can use that structure. And then you just press BLAST. And you'll get a list eventually of proteins that are similar. So we already have a list of possible templates for you. So we can go ahead and use those. So now we're going to want to download those structures from the PDB. So we can walk through with this 2RH1. So we would just go to the rcsb.org, search for 2RH1. It should be the first one that pops up. And we would download the PDB for this, so PDB format. I have those in the original file, so we're just going to copy them over for now. I think we also want to be in our template PDB directory. So you see that there's another directory inside there called original files. So I'm going to copy over 2RH1 into here. And we need to do a little bit of cleaning up. for this structure. So we're going to want to delete the residues listed, so residues 1002 and 1161. So we can open up our PDB, and this is kind of what it looks like. So let's find residue 1002. So here. And then we'll want to delete until residue 1161. So, alright. So you can do that manually in gedit or wherever you want. So you'll just want to get rid of those lines. And then save that. We're actually going to copy this over to our H1 isolated. Oops. Forgot one of my arguments there. Alright, so now you can see we have 2RH1, which we downloaded straight from a PDB. We have our isolated 2RH1. And now you'd want to do that for the rest of your templates. And also clean them up like we did these. However, we can, we can copy our already isolated PDBs from the demo folder. Alright, so now we have all of these isolated PDBs. So as you saw when we opened up the 2RH1, so we can do that again real quick, there's a lot of other information here, a lot of other lines, and Rosetta doesn't need all of that. So we want to make sure it's clean, there's no errors, that you don't get any errors that you don't know why they're happening later on in the run. So we're going to use the clean PDB script to do this. And it's pretty simple. You would just type in the script and then put your PDB file and the chain letter after that. Since we have multiple of them to do, we have a little, just some bash scripting or shell scripting to do this for us. So we can do this quickly. So now we have this list of PDBs file. Sorry about that. So now I just need to type in my path for Rosetta. So my path is going to be different than your path, but wherever it is for you. So you will not want to copy exactly this, but when you get to where the main directory is, it should be all the same. I'll have to scroll this over a bit. Alright, so let's just check. This might be more simple. So you can see the ones with the _A added to them are your new files that were made from the clean PDB. And these also should all be, the prepared files are in the demo folder if you have any problems with this or if you want to compare and make sure that they're all the same. And just a quick note, these were all chain A. So that's why they all have _A. But if you were getting chain B or chain H or something, depending on what it is, then you would want to put that in your argument for the clean PDB instead. And that would also pop up in your naming. All right, so now we're on to aligning. So we'll want to go to the alignment directory, alignment files. So we're going to use Clustal Omega for our alignments. So real quick, we're just going to cat our Fastas onto the screen because we'll use those for the alignment. So there's our target, and then going to our templates, and since clean PDB outputs those Fastas, makes it pretty simple, they're already there. So then you'll want to go to the Clustal Omega website, and you'll copy that in there. And then here is our alignment that we get. So you can download this alignment file. So this is what this would look like. You can just copy and paste this text into a file if you wish. So as we talked about in the presentation, your alignment is extremely important, and kind of you know what protein you'll be modeling, and you might have some background information about that. You might have predicted secondary structure or highly conserved residues that you know should be aligned, or residues that you know should be in the membrane. So you'll want to go through and look at your alignment and alter it for any of that background knowledge. In our case, we have an already prepared alignment file for you in demos, so I would just recommend copying that over into your alignment files. All right, so now we're going to want to get the span file, so the predicted regions that are going to be in the membrane. So I would go back to input files. Oh, I went too far, so I went out of my model, I think. There, now I'm where I'm supposed to be. So we're going to want to cat the full sequence. So Octopus definitely uses not just the sequences that will be predicted to be in the membrane, but it uses the loops and what those sequences are to also predict the membrane spanning regions. And in this case, if we were to put the altered sequence where we took out ICL3 and the N-terminus, then Octopus would actually only predict that there are 6 transmembrane helices. And we know, since this is a GPCR, there should be 7 transmembrane helices. So we're going to use the full Fasta sequence, and then we'll just have to go back and edit the residue numbering to make sure that that's correct. And you're welcome to put in the altered sequence and compare those results. So it's kind of interesting. So we'll want to go to Octopus. The website should be in the tutorial. You want to go to Home if you're not already to put in your sequence. So just copy, oops, I accidentally went back. Just copy and paste your Fasta sequence and press submit, and it's super fast. So you can see that the red regions are your helices, and there are 7, so that's just a double check there. So we'll want to copy this. And we'll copy this into a file called throughppl.octopus. And save that. Now there's a script that should convert an octopus topology file to a span file. So again, you'll want to type in your path to Rosetta. Alright, so let's open up our span file, see what that looks like. So here it's just telling us that there are 7 transmembrane helices, there are 400 residues. The helices run anti-parallel, so you have like the N-terminus region and then it turns, so if you were following it along, it would be, the lines would be anti-parallel. From the N to the C terminus, and then which residues are predicted to be membrane spanning. So this would be residue 34 to 54 would be in the membrane, and then 67 to 87, and so on. However, like I said, we're using the full sequence for this prediction, and we have our altered sequence. So residue 34 is no longer residue 34, and we also don't have 400 residues in our target sequence anymore. So you'll want to go through and edit those if you had to perform something like this where we needed to use the full sequence. So I actually have already that altered span file for you, so you don't have to go through and change that by hand. This will be much faster. So it's definitely what I recommend. So now we can see kind of the difference and what that looks like. So there's still 7 transmembrane helices predicted, but now only 280 residues. Now instead of 50 or 34, I think it was, we have 3. So you can go through, both of the span files are in the demos, the full one and this altered one. So you can go through and check that out and see the differences there. Now you'll need to define your disulfide bond, and that's pretty simple. We're going to have a disulfide between residues 72 and 150, and we have that prepared for you also in the demo file. So let's take a look at that. So extremely simple. You just put the numbers of the residues that are forming the disulfide. Again, I would say I would definitely recommend making sure that if you have to find some disulfides, that they're the correct residues for the residue numbering for your protein. So when you change the Fasta, you cut off part of the N-terminus, whatever is the first residue, Rosetta is going to number 1 as residue number 1. So just make sure that your numbers are correct from what they are. Don't just look up the disulfides and use those numbers without double checking that. So now we need to do the threading. So let's go back to our alignment files. So we have the 3PBL alignments.txt. Can't remember if we took a look at that, so we can do that real quick. Oops. All right, so here are the alignments. They look nice. And we have a script already for you that will make the separate Grishin alignment files already. So now we have separate alignment files for all of our targets, or sorry, our templates. You could do this by hand if you wanted, but this is much faster in this case. So now we'll need to do the threading. So we don't have a threaded PDBs directory, so we're going to make that. And then go into it. We're going to run the partial thread app. So you'll have to run this for each one of your template PDBs. So we're going to start out with 2RH1. So where in this command on your tutorial, it will say like PDB is capitalized, but you'll need to replace that with the PDB ID of the one that you're working with at the time. So this is an example, like I said, for 2RH1. All right, so that's done. And it says the output should be named 2RH1 out.pdb, and we can see that in our directory. So you'll want to go through and do that for all of them. For the sake of time, I'm also going to copy those over as they're already done. So in this, we want all of the PDBs out of there. Save them here. So now we have our threaded PDBs. So now we are actually going to run hybridize. We want to make sure we have all the necessary files. So we just created these threaded PDBs. Let's go into our input files and double check that we have those. So we have the Fasta, our weights files. We don't have the XML, so we'll still need that. Looks like we don't have our options file, and we have the span and the disulfide. So if you didn't have the weights file, we already have that. It looks like in our input files, but if not, you would just copy those over from the demo. And weights files don't typically change, so they're kind of part of the database. They've been tested and optimized. So unless you are an expert and have tested this, I would not recommend ever changing the weights on any for any of the score terms. So these are kind of how they're supposed to be. All right, so now we need to copy over our XML script, and this may feel like a little bit cheating to you guys that we're just copying this over, but I hardly ever start with a blank XML script, so I pretty much always start with something that's already been written. Either I wrote it or I took it from someone else, and then editing it. I think that's just the easiest way to go about that. So let's take a look at what that looks like. So we have our score functions defined up here, which we kind of saw in, sorry about that, we saw in the presentation. So we have our stage 1, membrane weights, stage 2, stage 3, and then our full atom membrane high res mem smooth, and we're going to call that membrane. So then we have our options for hybridize. So we set all those here. We say which templates we want, which score functions we want to use for which stages. We'll want to clear the constraints before we do our fast relax step. And then the movers that we want to run, we put them in the order we want to run them in protocols. So add, hybridize, clear constraints, and relax. And then which score function we want to use for the final score file output. So all of that seems in place. Now we're going to want to copy over our options file. Whoops. All right, so this is our options file. We're kind of breaking it up into input and output options, membrane options, relax options, and then just reducing the memory so that none of these fail just for memory reasons. And a lot of these are, I would say, definitely recommended options for running CM. So if you want to change these, I would look into what they do before I would just go deleting these or changing these options. All right, so now I think we're ready to run hybridize. So let's go back to just the My Model directory. Real quick, let's make a directory for the output files so that they're not all in the same directory where our other stuff is. All right, so again, your path to Rosetta. Then we want to specify our options file. Actually, I think, let's scroll over. I think I missed a step there. No, it didn't. Okay. All right, and if you press enter now, all of your output will go to the screen. And you won't be able to do anything on this terminal. So I would recommend actually saving this to like a log file. So I'm just going to call it RosettaCM.log and then a little ampersand so it runs in the background. So we can submit that. That should take around 30 minutes, give or take. So we can let that run. And this is only going to generate one model. So you can change, let's see if it's in your options file. Yeah, so you have the instruct option here. So we have this specified to 1, but you can change that to however many structures you want to output for each run. So if you wanted 50, then you would just change that 1 to 50. And then you have some final models already created for you, so you can kind of see what sample outputs would look like. So if you go into demos, you have models in your output files, and then you also have some of the final models that we have selected out in the final models directory here. So I would encourage you guys to look at those. If you have a lot of time, you could run maybe some more instead of just one with your run and see how they turn out and how they compare. We should be available, I believe, via Discord if you guys have any questions, or feel free to email me to ask me questions about this protocol. And thank you guys.